So neoantigens are for most part mutation-derived antigens that are expressed solely by the tumor cells and are absent in healthy cells. So because these antigens are so foreign to the self, the T cells that are reactive against these antigens have the potential to be highly cytolytic against the tumor because they were not subject to central tolerance. Having said that, despite the presence of these highly immunogenic neoantigens in the tumors still progress, which means they evade immune surveillance, and that happens because the tumors can express very strong immunosuppressive programs and cause immune dysfunction. So the PGB-001 program was designed to predict personalized neoantigens, generate the vaccines, and administer the vaccine in a minimal residue disease state, or in a state where the tumor did not exist anymore or was as removed as possible, so that the immune suppression caused by the tumor is limited and there's a chance for the T cells to react to the vaccine. And there are several neoantigen vaccines that have been administered and in different trials and different projects, but sometimes the data from these trials is a little skewed because the inclusion criteria is really limited. For example, they would only focus on a certain tumor type or patients that have a certain HLA expression or a certain tumor mutational burden. But in this trial, we kept the inclusion criteria really broad. So we enrolled patients with different types of solid malignancies as well as hematological malignancies. The patients were not screened on basis of any kind of HLA expression, and they were not screened on any threshold for a tumor mutational burden. And all the patients were treated after either surgical removal of the tumor or in case of hematological malignancies after stem cell transplant. All the patients had a very high chance of tumor recurrence. So when we vaccinated the patients, our overarching goal, this is a phase 1 trial, so our main purpose was safety and feasibility, but the overarching goal of the program is to be able to establish a strong neoantigen-specific effector and memory T cell response so as to either prevent disease recurrence, delay disease recurrence, or in some cases, make the patient more receptive to receiving other immunotherapies.